learned that I always enjoy talking about and learning about regeneration. So
yeah, no, I guess we have Bernice Grafstein here, so it
yes, of course, the Bernice Grafstein. And I wanted to say hello, Bernice. Nice to meet you, albeit via Zoom.
I think you're muted.
Oh, I think you're muted, Bernice. The microphone icon should be in the bottom left.
Okay, I think am I live now?
Hi, Bernice.
Can you hear me now?
Yes, yeah,
I
pleasure.
just want to say hello. That's all I'm going to say.
Well, we're really happy to have you. So that's
Yes.
one of the unexpected benefits of being able to do this over Zoom. So I guess we should count our blessings then.
Yeah.
So I'm a great fan of Zoom. I think it's wonderful.
This
You can get lots of people from all kinds of places.
That's right.
Yeah,
it served us well through these wild times.
Do you travel a lot now?
I try not to, but I'm selective with my travel. I haven't been to Europe since before the pandemic, but I occasionally make it to the East Coast and up and down the California coast. But I find that with Zoom, well, obviously I was supposed to be out there in person, but the flight was moved and then canceled, or canceled and moved. I try to limit my travel to once a month, if possible. Is that a lot? I don't know if that's a lot. That's used to be before the pandemic. I was traveling all the time.
Yeah, it feels like a lot now, but it wasn't a lot before.
Yeah. One of the advantages of podcasting is that you have to be in a chair recording once a week. And so it keeps me close to home, which is good. I had a dog for a long time, but I had to put him down after about 11 years.
Oh, no.
And yeah, Costello, he reached a ripe old age for a bulldog, but I'm warming up to get another dog. I'm obsessed with coonhounds now because of their olfactory ability, the opposite of a bulldog who has essentially limited olfactory ability. So soon I'll have puppies, and then I plan to travel very seldom.
Yeah, I shouldn't even say this. We went to Japan for a week, and we came home and my cat had redecorated the house considerably.
Wow. Oh yeah, cats are great too. I just think the dog thing kind of got under my skin, and I can't seem to be long without one now. I don't know what it is. I think they hijack the neural circuits for human attachment, and
they are symbiont, right?
Oh yeah, I'm convinced that there are not unique circuits for different forms of attachment. Obviously, there's different forms of attachment, of course, but I think that there's one system, and I think that explains a lot of wonderful things about life and pathology about life. But the great thing is a dog is universally devoted to just having a good time. Right, that's all they really care about. Bulldogs are a remarkable species because they are very parasympathetic dominant. I mean, a nuclear bomb could go off and my bulldog Costello might move his eyeballs, but it's pretty terrific because that's not my personality type. And it was really wonderful to see that an animal could exist in the world so peacefully despite everything. So
doesn't read the newspaper, clearly.
What's that?
Doesn't read the newspaper.
No, doesn't. And even if he could, he wouldn't care.
He has better uses for the newspaper.
Yeah. The last point. Yeah, exactly. The last point about this is that dogs have a really amazing ability, bulldogs, that they essentially look disappointed all the time. And then when you give them something they like, they look delighted for about a nanosecond. So pretty soon they have you operantly conditioned to try and please them. It's I don't think they know they're doing it, but it certainly works. It's pretty delightful, but it kept me working hard.
Well, you got quite a big audience now, Ed.
Yeah, I
Over
think
100.
it's about time to start. It's been 5 minutes, so we'll probably have a few more people join. Before I proceed, I just want to confirm with everybody that it's okay that this be recorded. Andrew, especially you. We put it on our lab website, not lab site, IPN website. That was one of Bernice's wishes, in fact, that these be shared. So I think that's a nice sentiment and hope to be able to do that. Great. So just got a message, the people who aren't registered for, yes, so people can join. Okay, so let's proceed. If everyone can see my screen. It's really a great pleasure to have this opportunity, although it's not quite the way I expected it would come out. I think it's still going to be a great experience. We met with Andrew this morning and, or I guess this afternoon here, his morning, and it was really a wonderful experience. He's a uniquely empathic and thoughtful individual, and I think that ability that he has to do science communication and to talk about things in a way that just makes sense, that is reflected in the way he does science as well, this very clear, organized thinking. So I think everyone's going to really enjoy the talk today. A little bit of introduction in the beginning. So unfortunately, there won't be a wine and cheese today. That was one of the things that was sacrificed as a result of Air Canada's negligence. Let's see. Now the question is, how do I advance my slides? Okay. Can you see the next slide? So I wanted to start out just by thanking some people who have made this series possible. We've now been going for a number of years. The first person who I think I should recognize is in addition to, of course, Dr. Grafstein, is Sal Carbonetto, who retired a few years ago, but Sal was really instrumental in bringing Bernice to our attention and connecting us and making sure that this really great series came to fruition. So I still want to acknowledge and thank Sal. At the time, Joe Rochford was the director of IPN, and so together they really made this happen. So I want to acknowledge both of them. In addition, the Neurodevelopment Office was responsible for setting up this, the fund, which is now allowing us to do this, hopefully in perpetuity. So this has now become one of the things that we can look forward to every year from the IPN, which is, a chance to bring in great scientists to talk to our community, but also to talk to the neuroscience community at large, so that people can enjoy and see what's really motivating all of us, the great things about neuroscience. And then the last person I wanted to acknowledge is Adriana Scambettera, who is a recent addition to the IPN. She's the project manager, and really it was her hard work that created the multiple incarnations of this seminar today. Unfortunately, it may not have turned out exactly the way she had dreamed it would be, but I think it's going to be wonderful, and we're all grateful to her for the work that she's put in to set it up. And of course, most importantly, Dr. Bernice Grafstein. Just a little bit about Dr. Grafstein. I don't want to embarrass her since she's here, but Bernice is the professor of physiology and biophysics and the Vincent and Brooke Astor, a distinguished professor in neuroscience at the Weill Cornell School of Medicine. Bernice is originally Canadian. She has her BA degree in physiology from the University of Toronto, and she has her PhD in neurophysiology from McGill University. So she has a long-standing relationship with the university, and in particular in neuroscience at McGill. Following her work here, she did a number of really impressive, her work here actually was impressive, and she's continued that throughout her career. She initially had a faculty position at the Rockefeller University and shortly thereafter joined the faculty of the Weill Cornell Medical School in 1969, where she's been since. In terms of the work that Bernice has done, some of it is really classic seminal work, the characterization of the mechanisms underlying spreading depression in the cerebral cortex that, is textbook material. But and Bernice has always had this sort of physiologist's approach, but she's actually contributed enormous amount to our understanding of neuroanatomy and regeneration. And so she characterized neuronal connectivity. She studied neuronal connectivity under Victor Hamburger at Washington University, worked at Woods Hole. Bernice has done an enormous amount of work on the regenerating axons in the goldfish optic nerve, and some of that led to work on that has basically won her accolades in terms of understanding regeneration and recovery from injury, and how do nervous systems respond to injury. There are cellular changes that occur that are very important. Something that for me is of personal interest is that she's the person who's responsible for, as part of her studies of intracellular transneuronal transport mechanisms, she showed that it's actually possible to inject a radioactive amino acid into one structure in the brain or in the eye and have it transport transsynaptically to label structures multiple synapses away. So she initially, I think, showed it in the geniculocortical projection, which is something that I did my PhD work on. And so I'm actually couldn't have done that work without her initial discovery. So she's really made a number of contributions that have continued to benefit the community. And as a result of all of these contributions, and because of her ongoing work, to be honest, and her service to the neuroscience community, Bernice has received a number of accolades. I think she was recently elected a member of the American Academy of Arts and Sciences in 2021. Bernice, in terms of service, she was quite early on one of the early Society for Neuroscience presidents. I think she was the first woman president of the Society for Neuroscience. And of course, in those days, it was a much smaller organization, but I think that it's because of people like Bernice that it was able to grow to become this kind of international behemoth. She subsequently received the Mika Saltpeter Lifetime Achievement Award from the Society for Neuroscience, which is an award for, in particular, a woman who has contributed a lot to the advancement of neuroscience careers, especially of female scientists. But I think Bernice has always said that it's really what matters is the science, not the scientist necessarily. So she's really somebody who's focused on the just the beauty of science. And in particular, I think physiology. She's a life trustee of the Grass Foundation and has played an important role in training Grass Fellows and making sure that people have opportunities to learn about science, including this annual Grafstein Lecture that we have here. So thank you, Bernice. We really appreciate what you've done. So I'd like to now, and I guess it's kind of awkward with Zoom. I'd like to, if people want to applaud in some form, I think it would be appropriate, but thank you very much, Bernice. Okay, so the next thing I'll do is hand over the podium to Pierre-Luc Rochon. Pierre-Luc is a PhD candidate in the IPN who works in the lab of Arjun Krishnaswamy, studying the kinds of things that Andrew Huberman actually has played an important influential role in as well. That is the wiring of the retina, and in particular dendritic, in the case of Pierre-Luc, laminar connectivity and specificity, but importantly, how the decisions of who connects to whom and the structure of retinal ganglion cells is determined. So Pierre-Luc, I'll hand the podium over to you.
Thank you, Ed. All right, so it's a pleasure to introduce Dr. Andrew Huberman as our invited speaker for the 2022 Grafstein Lecture today. I'm just going to go where he came from so that we have a good idea of where we are today. So Dr. Huberman first started by receiving a bachelor's at the University of California in Santa Barbara, and followed with a master's from Berkeley, where he got interested in roles for sex hormones or androgen in early development. This sort of pushed in to follow up, where he worked in Dr. Barbara Chapman's lab afterwards at the Center of Neuroscience at UC Davis, when he focused on the development of retinal geniculate projections in ferrets. And more specifically, what he did is sort of express FAs in ferret retinal ganglion cells, or RGCs, through electroporation, and sort of studied the consequences of their axon patterning in the primary visual thalamus, or what we call the LGM. Investigating sort of this interplay between the genetic and the activity patterns and how these pattern retinal inputs into a really important anatomical link between the eye and the brain. Now, for his postdoctoral studies, he turned his attention a bit away from the RGC axons or the projections and turned inwards towards the dendrites, sort of developing some of the first mouse lines that grant access to retinal ganglion cell types in Dr. Ben Barris's lab at Stanford. And he sort of followed this interest in RGC, starting with sort of an independent position at UC San Diego afterwards, pioneering like really important genetic tools that sort of, as Ed pointed out, that we really use. So I and other grad students that are interested in the visual neurosciences, we use a lot of these tools, and also some of his papers are sort of the foundations for our graduate projects. Now, in 2016, Dr. Huberman moved away from, moved back into Stanford University and extended his study of retinal ganglion cell to learn how they become altered in glaucoma. And this, of course, all of this works has awarded him a lot of prestigious awards, starting with the Helen Hay Whitney Fellowship, notably in his postdoc. Not only that, he also is a scholar of the McKnight Foundation, the Pew Biomedical Foundation, and was selected for the Catalyst for a Cure glaucoma research team in 2016. And not to mention that he has a growing profile outside of academia, where coverage of his work is done by Scientific America, Time Magazine, and of course his top-ranking podcast on Spotify, the Huberman Lab. Now, in his most recent works, it seems to involve more back to the central projections of the retina, but on more non-image forming brain regions involved in fear and anxiety, which was notably published recently in Nature. And judging by the title of today's talk, I have a feeling we're going to learn a bit more about that work. And with that, I want to give it back to Dr. Huberman, and we look really forward to hearing about your work.
Great. Well, thank you so much for the wonderful introduction. And it's really a delight to be here speaking with you. A brief apology that there were some flight cancellations that prevented me from being there in person, but hope to do so in the not too distant future. Today, I'm going to talk about interactions between vision and autonomic function, something I've long been interested in, but that only in recent years have I had enough sort of energy and bandwidth to focus on. I'll just very briefly, out of an inability to suppress myself, just mention that what we're finding in regeneration. I won't show any data, I'll just mention it for Dr. Grafstein and for others who've done the pioneering work there. I mean, so much pioneering work on regeneration out of the labs of our Canadian neighbors, right? It seems very clear that's the way it's gone historically. Basically, what we're finding is that we know from development that neural activity plays a key role in the preservation of cells and avoiding naturally occurring cell death. We also know that their innervation with their targets is critical for maintenance of cell health. What we find, and this is in the mouse model, but what we find is that increasing neural activity after an axonal lesion, either by visual stimulation or by chemicogenetic or other manipulation, can encourage the survival and regeneration of ganglion cells. That perhaps is not so surprising, given that most of regeneration seems to be an attempt to recapitulate development in some way or another. The more recent data, which are not published, and I'll just mention, and hopefully we could talk about sometime soon, is that we're now finding that if we increase the activity of the targets, the central targets from which those ganglion cells are divorced when they're injured, that activity can serve as a kind of a beckoning role for the axons. In other words, activity within the targets is important for allowing regeneration as well. The spatial range over which that occurs and the specific molecular cues and whether or not it's growth factor related, et cetera, isn't clear, but we're excited about those results. And the last point I'll make about that is that we're excited about them because the clinical data, mostly out of labs in Germany, because this is not an FDA approved procedure, but the clinical in the US, but the clinical data show that for people who have damage to the retina, that giving a stimulating halo of sorts to stimulate, believe it or not, action potential activity in the visual cortex and geniculate, presumably also, can serve a preserving role for remaining cells in the retina. So the point here is, that if there's signals that propagate from the ganglion cells to within the ganglion cells themselves that allow them to regenerate to some extent. These aren't, a total regeneration as you might see in the goldfish or in an amphibian model, but there are also signals emanating from or required from targets to beckon incoming axons after injury. And so we really need to think about it as a pre and post-synaptic dialogue rather than, for instance, the idea that just the ganglion cell is responsible for its own regeneration. So I'll leave it at that. And if you have additional questions in the Q&A, we can certainly talk about those. I'd even be happy to put up some data if we want to do that. But for sake of time and for sake of focus today, I want to talk about visual autonomic interactions. And my interest in this is several-fold. And then I'll put my slides up. But just to frame this, I've always been interested in the visual system. We are such visual animals. People with low vision or no vision can learn to navigate the world, of course, with canes, with seeing eye dogs. Outsourcing vision is still probably the best way for a completely blind person to navigate the world with a combination of cane and seeing eye dog. There are emerging therapies where people wear glasses and then they have a dispatcher who views the scene for them and gives them instruction. They actually prefer that less to the more tactile signals of dogs. Pretty interesting to think about for a lot of reasons. But we're so visual, low vision, no vision folks can manage, and often do quite well and live very active, thriving lives. But we're so visual. And having spent a number of years focusing on the different cell types of the retina, the ganglion cells in particular, the ones that connect the eyes to the brain, I've always been intrigued by this idea that, you see certain things and they trigger an emotional response, and you see other things and they trigger no emotional response. And then, of course, I wanted to avoid getting involved in the study of emotions because there's still a dialogue as to what an emotion is. And for a biologist like me, that's a scary discussion. And I figured I'm aging at a rate that made me want to bypass that discussion and simply ask, well, what are the interactions between vision and internal state? And internal state is a lot of things, right? Happy, sad is sort of emotional. Are you feeling peaceful? Let's just make it very simple and say vision and whether or not you become more alert or less alert. And there's a system that's well known to control that, which is the so-called circadian system, or these cells within the eye, these non-image forming so-called melanopsin cells that basically adjust our internal state to be more awake when it's sunny out and more asleep when it's dark, to adjust to the circadian cycle. So before I hit the first slide, I just want to, pay, acknowledgement to the fact that the relationship between vision, although it's non-standard vision, and internal state is a powerful one that's highly selected for in every mammalian and probably other species as well. And the idea that light arriving at the retina and then transformed within the eye and communicated to the brain would control the internal state of a person or mouse in a profound way is not a heretical idea. But what I wanted to understand was how this could occur on faster timescales, not on circadian timescales, not on the order of hours, but on the order of seconds and milliseconds. So that's what we're going to talk about today. And I give you that long preamble because I want to make sure that when I say internal state, or I say the word fear, or I say the word confrontation, I want to acknowledge at the outset that, we have no idea what animals are feeling. In fact, most of the time, we don't know what people are feeling. And most of the time, we don't even know what we are feeling. Language is not a great tool to explain our internal states. But with all of that said, I'm going to move here to a shared screen mode. And then I'm going to share my screen, and hopefully, not because I wouldn't be happy to share my home screen, but hopefully the only thing you see is my presentation as opposed to my silly notes to self. Okay, great. Great. So today, and I promise not to go over so that we have time for questions. I may speed up a little bit here and there, and please let me know if you have questions. Today I'm going to talk about visual and autonomic, right? Visual and autonomic circuits controlling stress modulation. And what I want to do is first do a little Gedanken experiment, little thought experiment. So let's say you were walking through this field and you were unconstrained in the sense that you could walk, you could run, you could take a nap, you could have a picnic. My guess is that you would just kind of stroll around, hopefully not looking at your phone, but just kind of ambulating around or through it. Let's say then you see this object in the distance, or let's just, and feel free, of course, because this is unique. I've never been on safari, but let's say that you were walking down a street at night and you don't expect to see anyone, and all of a sudden you see a person, right? So you could translate lion for person. So there is an optimal adaptive response to this, right? If to a threat, potential threat in the distance, which all animals, including you, would show, which is to stop, to pause, right? There will be a shift in your internal state and you would adjust your behavior almost with certainty. Now, even a slight shift in your visual perception could alter what is now an adaptive response. So why would you pause? Well, as we'll soon see, all animals, when they detect a threat, but they are uncertain if the threat detects them, they freeze or pause. And when I say the word freeze, it's a little bit misleading because a lot of people think of freezing as panic or being frozen. But actually, as we'll soon see, and I'm giving away a little bit of the conclusion here, the freezing response is the lowest level of autonomic arousal, the lowest, okay, in humans and in animals. And we do experiments on both in our laboratory. So a lot of people don't realize that. They think when you freeze and, paralyzed in fear, it sounds so catastrophic, right? But the freezing response is actually very adaptive because it allows you, for instance, to just see if this animal would just leave without making them aware of your presence. Now, let's say the animal looks at you. Now a lot changes, right? So now it's a question of, does it see me? Well, likely it does, especially in the daytime. Now you have options, which may or may not be a good thing. Do you pause? Do you continue to pause? Well, that's one option. The animal could then turn its head and go in the opposite direction. Or do you move, in which case you are more detectable, but at the same time, you make yourself, so you're higher probability of escape, but you make yourself more detectable. So you're playing you're gambling here, right? This is an optimization strategy. Okay, this has been modeled extensively in animals and humans, and we'll talk about that. And then, of course, if the animal is right upon you, the freeze response is not great as an adaptive response. This is the sort of fight for your life opportunity. And we can talk about how this photo is actually taken. This was a real world attack circumstance. This photograph was not taken by me. It was taken by my friend Michael Muller. And it's a very interesting thing. The other thing you'll notice is look at the pupils of this animal. Even though the pupils are, this one looks quite small and this one, there's some asymmetry here. There's a bright flash, but were there not to be a flash, the pupils of this animal will be very dilated. There's a system of autonomic arousal that impinges back on the visual system. We'll return to this later. The visual system is feeding the circuits of the brain involved in arousal, and the circuits of the brain involved in arousal are feeding the circuits that serve vision. In fact, under conditions of arousal, assuming constant luminance, the pupils get bigger, which actually changes the visual field so that you see certain things in bolder relief with more detail, but you don't see as broadly in the visual scene. You basically have different eyes depending on whether or not you are alert or calm. Not incidentally, law enforcement knows that if someone has pupils about this big, that they are likely on some sort of stimulant, amphetamine type drug. And if their pupils are itty bitty small, that they are likely on some sort of sedative or narcotic type drug. Very, very consistent set of findings. There's also a luminance-based change in pupil reflexes that interacts with that, which is also not incidental. Luminance tends to drive arousal. We'll talk about this. Okay, so sorry, this puts a menu at the top that makes it hard to see my own slides. But so I'm not going to read my own slides. But so what we're essentially trying to do is to link vision and emotions. But what we're really talking about is autonomic functions in the brain and body. And, you know, I'd like to discard, even though I'll use the terms fear, et cetera, but nervous, worried, scared, fear, afraid, jitters. You know, I'm so grateful I didn't become a psychologist, because I think I would have spent most of the time obsessing over operational definitions. You know, so much of the miscommunication and maybe even misinformation we see in the world these days around science is because of the lack of a common nomenclature often. It's so hard, misunderstandings are so frustrating. And so I'd like to really clear up any misunderstandings from the outset. We have no idea what people feel. It's very hard to measure. We can ask them, but what we can do is measure things like breathing rate, heart rate, pupil size, pupil dynamics over time. We can get a sense of their so-called autonomic arousal, the circuits that they don't control directly, but that reflect shifts in internal state from those of more alertness and readiness to states of more drowsiness and calm. It's a continuum. And we could probably even think of it more like a seesaw, because the autonomic nervous system essentially consists of the sympathetic nervous system and the parasympathetic nervous system, and they are in a push-pull. Were we to divorce your heart from certain brain structures by, say, cutting your vagus nerve, your heart would beat very fast. It would still beat. In fact, much of the greater work in the last few years, or I should say the stronger work in the areas of neuropsychology of depression and neuroscience of depression, has shown that the prefrontal cortex is involved, for instance, in a depressive episode or a depressive loss. People will reframe the loss as, well, we had a great years together. There are signals from the left dorsolateral prefrontal cortex, left in particular, that flow down the vagus nerve to slow the heart down. There are other ways to slow the heart down, but that seems to be an essential part of the reframing and calming response to what would otherwise be an anxiety, panic, and depressive response. So physiology and emotions are linked, and we can look to physiology in very reliable ways. Now I just want to make sure I can advance my slides in very reliable ways. There we go. So these are just this is just a gallery of different images, each one designed to evoke a slightly different, quote unquote, emotional response. So this hopefully evokes a different emotional response than that. This is no big deal unless you're afraid of honeybees. So there's context. This is just kind of cute. See, I've always wanted to own one of these little guys. I don't know if they make good pets or not. This is adorable. You sort of have to, I mean, I think, I don't know this to be a critical test of sociopathy, but if you can't appreciate how adorable a baby elephant is, I would wonder. It's pretty cute. And then, you know, nuclear war, terrible. Like, so here's the point. We are very good at looking at an image and extracting some sort of emotional value or valence from it. And so we can think about autonomic arousal combined with valence, right? Whether or not something is deemed good or bad by us. And then that, of course, is molded by context, right? I mean, for some people think these are beautiful animals. I don't like snakes at all. I don't mind spiders, actually, at all. But I don't like snakes. I don't have a phobia, but I don't like them. I don't like dark spaces where I can't see anything. So, you know, we tend to have strong emotional / autonomic responses to these things. So where does that happen? I've been working on the visual system for about 15, 20 years. I know there's an amygdala to visual cortical connection, but you lesion that, not a whole lot happens. So where's all this stuff getting integrated, and how is it getting integrated? That's the question. So what are the neural circuits that integrate vision and internal state to drive basic adaptive behaviors? That's where we started. And the main person working on this in my lab was a graduate student named Dr. Lindsay Saleh, who's now finished. She's doing her postdoc at Caltech with Dr. David Anderson, who works on a number of things similar, but far more vast than the kinds of projects we take on in our lab. And I should say, prior to Lindsay's arrival, a postdoc who now works on the human aspect of all this in my lab, which is Dr. Melise Balban, who was a graduate student with Marcus Meister and developed a lot of the behavioral assays that I'll talk about. So I was very fortunate that the two of them converged in the lab at the same time. Lindsay decided to explore some of the circuitry of autonomic or visual autonomic interactions. Melise was responsible for developing those assays as a graduate student, although Lindsay expanded on them. And then Melise decided to work on humans in my lab. So we all know that the retina is where visual information comes into the brain and is communicated through the classic dorsolateral geniculate structure, which in a mouse just looks like an egg. It's not very sophisticated in its shape, but nonetheless, that information is relayed to the visual cortex. This is, textbook stuff for most of you. You already know this, but nonetheless, there are a bunch of other pathways too, including pathways to other subcortical structures, the circadian clock, the areas of the brain involved in pupil reflexes. Over 46 central structures receive direct retinal input and probably more. Most, I should mention, by collateralization, not one-to-one connection, by individual ganglion cells branching to multiple targets. There's a lot of redundancy. You have targets involved in reflexive eye movements. I've spent most of my career digging around in here in the subcortical visual pathway. I initially started to do that as a graduate student because no one else seemed to want to work on the DLGN. Everyone was working up here at that time. There were a few of us. There were a few people working in the retina hypothalamic tract, but I've enjoyed trying to parse what some of these more mysterious structures are and how they work, mostly because I don't like crowds. I would have liked to be there with all of you today, but I'm a bit of an introvert when it comes to my behavior and my scientific choices. I don't like crowded territories. And I also believe in the deletion test. I think that if someone else is going to do something pretty well, I try and work on something else. It's not that I'm not competitive. It's just that I'm not competitive enough to want to do what someone else would do, just not for me. So I've loved digging around in these various targets. We also know that nearby are all these other brain areas associated with so-called fear. And really what they are involved in is aversive behaviors and prosocial behaviors, or shutting down prosocial behaviors, or autonomic arousal, or pain, or all the things that are associated with the kind of interactions that would lead to mating or fighting or fleeing or these kinds of things, or sleeping. The areas of the limbic circuitry and its associated circuitry are really about autonomic arousal first, and then about specific behaviors. Because if you think about any behavior like mating or not mating, it's either an approach or a retreat. You think about sleeping, it's a decision to stop and be quiescent for a while. You think about food and the pursuit of food, there's mobilization required. So again, I've always thought of myself as not the most sophisticated thinker, and I don't like it when people disagree merely on the basis of lack of common nomenclature. So I said, okay, I'm pretty sure everyone understands that alertness is required for certain behaviors, and maybe more alertness for certain behaviors than others. And then calm is required for certain behaviors or for the lack of certain behaviors. I think I can work with autonomic arousal. That seems more tractable to me than trying to parse like what a mouse or a person is thinking when they see something. Anyway, we'll see if it was. And then this has always been a little entertaining to me, which is that very slight shifts in, if you didn't catch that, in visual stimuli. Like if you look at the statistics of this image, and then you look at the statistics of this image, they're not that different. I mean, they're different enough, and you have a recognition that the one thing is kind of cute and silly, and the other would be terrifying. And that's incredible. Now, in the real world, there would be size and context, et cetera. But to me, that's incredible, because what it means is that it's fast, it's meaningful, and that also the circuits are likely to be hardwired in some sense, or at least present by the time we know what the difference between a kitten and a lion is. So without further ado, this is the model. So what are the neural circuits is an interesting question, perhaps. Like, can we link the vision? Where do the limbic system and the visual system interact? We'll put on our anatomy hats and play that game. But we can also ask, how does this actually shift internal state? Maybe we can figure out the difference between arousal, motivation, drive. I mean, these are the high-level concepts that we'd all like to understand. Maybe we can understand that, maybe not. There are a lot of arrows here. It's likely to be complex. State drives perception. Perception drives state. It's not unidirectional. Okay, so then we need a real world model. And then this is the real world model, which is that animals like mice tend to be preyed upon by animals like raptors. I unfortunately didn't take this picture, but this has been translated into a laboratory paradigm. This is some beautiful imaging. This, I think, is an owl actually coming in to retrieve its food. And so this is the model that was developed, behavioral paradigm that was developed by Melise Yilmaz when she was in Marcus Meister's lab, and then Lindsay Soleil and I and many others now have used it, the so-called looming assay. So here's what it is. You have a top view over here. There's a little shelter, a little hiding place here. It's a screen above and a mouse in this chamber. We'll just play this. The mouse ambulates around. The looming stimulus comes on. The mouse freezes momentarily and then runs into the shelter. Very consistent. They require no learning to do this. They require no visual cortex to do this. They do require vision to do this. If you put an overhead disc that's on a black background expanding white, you don't see it. If you put the stimulus on the side, you don't see it. This is, we think, mimicking an incoming predator in some way, some reflexive response. The other reflex that's very consistent is the falling reflex, where animals essentially will look up when the visual world is moving up, we'll look down, excuse me, when the visual world is moving up, but that involves some vestibular changes, so it's a little bit tougher. Now it's not easy to see, but you will see it. There is something that we see pretty consistently in about 20% of animals, 12 to 20%, that once the animal runs into the shelter hide, it'll take its tail and rattle its tail, which, again, we don't know what the mouse feels, but is thought to be related to aggression. The one thing we know is it increases the salience. It would be like me running and hiding from that lion or someone who wanted to harm me, and then hiding under a rock and saying, "I'm over here," okay, which is a little strange. But here's how it goes. So you'll see the tail flick against the wall. I've got this on loop. And later I'll show you that it actually makes a sound. So they run into the shelter. They may be signaling to conspecifics. There's some really nice work. It's done by a couple, Blanchard and Blanchard, husband and wife research team out in Hawaii, that have hypothesized what this might be about. But we can talk about that later if you like. So the other response that looming stimuli sometimes cause, and it's somewhat random, later I'll tell you how to predict which one will occur, is a freezing response. So here's a mouse, just kind of checking out this arena, doing what a mouse does, and then boom. And if I didn't show you that looming stimulus, you might think I paused the video. I mean, it's just, now this is actually important in some ways. The freezing response is a behavior. It's a lack of behavior, but it's a very active response. It is not easy to stay that still. Think if you were ever playing hide and seek, right? Or in the horror movie where somebody's hiding in a closet and they're trying not to move or breathe too loud. So the freezing response is a very active behavior. I think we need to think about it a little bit differently than a passive behavior. Very interesting patterns of motor neuron activity are required for this. So we can quantify this through these actograms. We can do this. I'll show you a bunch of different ways of viewing data like these, but there are 15 different animals on the left. You can see some animals, they run, then they hide, some freeze the whole time. Those little squiggles are tail rattling episodes. So some animals tail rattle, some don't. You'll soon see that they always, if they tail rattle, they always do it within the confines of the hide. They hide and then they go, "You didn't get me," that kind of thing, or something like that. Again, we don't know what they're thinking. So we want to know, what are the neural circuits that integrate vision to drive internal state, to drive internal adaptive behavior, excuse me. So at the time that Lindsay did this, now there are additional methods to do this, but what she did is she exposed animals to these looming stimuli, and then she stained the brains for CFOS, which is a marker of immediate early gene activity, of course. And she did a differential screen of the brains to see what areas were activated in the looming exposed versus non-looming exposed, as well, as animals that were exposed to other kinds of looming stimuli, like the bright looming stimulus that doesn't evoke freezing. And she centered in on a pretty interesting brain area which is the ventral midline thalamus, or VMT. The VMT is a dense cluster of nuclei. I teach neuroanatomy to medical students, and I know we have other expert neuroanatomists, for instance, Dr. Grafstein here. So I'm open to revising my understanding of the midline thalamus, but the midline thalamus is a kind of a no man's land of unidentified or poorly identified structures compared to other areas of the brain. You have this nucleus reunions, RE, a bilateral structure here and here. You have the xiphoid nucleus. You have the central median nucleus. You have a bunch of other nuclei in and around this area. And I think it's fair to say that the VMT is 3 or 4, maybe 5 or 6 different nuclei, including a nucleus that lies dorsal to all of this, which would be here, these little bat ear looking things at the midline. So here's midline thalamus. This corresponds to this region, which is the habenula. And I'll just tell a little anecdote about the habenula. When I was a graduate student, an undergraduate, someone asked in neuroanatomy, what does the habenula do? I had a wonderful anatomy teacher, a guy named Ben Reese, who's a retinal biologist, a phenomenal neuroanatomist. I adore Ben. He's a very nice person, very, very rigorous. And he said, we have no idea what the habenula does. And he was right. Now, in 2022, we know that the habenula is involved in suppressing dopamine responses under conditions where the outcome is what you didn't want. It's more or less associated with disappointment and has become an interesting structure for sake of understanding depression. And I should mention those data are true in zebrafish and in mice and in humans. So the habenula is interesting, is in the midline. The nucleus reunions and xiphoid and these other structures are in the midline. And it suggests that the midline is playing somehow a modulatory role. Now, I don't want to get too far into the aspects of neuroanatomy, but my hope is always that, if the internet goes down or something, which hopefully it won't, that there'll be some takeaways that are worth value along the way. And one of them that I learned early in my career from a guy named Harvey Carton, who is a friend and an amazing neuroanatomist, and is the following. When you learn spinal cord anatomy, you learn that the structures in the ventral horn are motor neurons, right? They're motor neurons. Motor neurons are down here. And then up top are the sensory neurons. And then around the midline are the interneurons that modulate all the other stuff. There are exceptions, but that's largely true. Then you migrate forward into the thalamus. And this is where I think Harvey really opened my eyes to something, which was the same is true for the thalamus and for the midbrain superior tectum. If you look at the ventral structures, they tend to be motor nuclei. You look at the dorsal structures and they're sensory nuclei, like dorsolateral geniculate nucleus. Now, in the primate, the dorsolateral geniculate nucleus gets moved down to the base of the thalamus. So when you look at it in a coronal cross-section, you see at the bottom, but it started embryonically up top. And guess what? All of the midline structures in the thalamus are modulatory. So from the tail of the nervous system, meaning from the tail of the spinal cord until you get into the front of the nervous system, there's a principle, which is that the sensory neurons sit atop, the motor neurons sit ventrally, and the modulatory structures sit around the ventricles and midline. And the story around this is a beautiful one that relates to fish and amphibia, which is the proximity to the central canal and to the ventricles actually gives those neurons access to some of the more slow changing chemical milieu of the body. And this is, I think, a really interesting way to think about midline thalamus is it's has access to sensory, it's at a position to act as a hub between sensory motor integration, but also to understand the chemistry of what's happening in the periphery. Changes in acidity, for instance, changes in adrenaline released in the periphery. Fascinating way to think about this stuff. I take no credit for that. That was all Harvey. And it was like a big duh and a big wow. I sort of forgot that what's true in the spinal cord is also true in the brain. And it also raises an interesting question, which is what is all that cerebral cortex doing? Because there it's both motor and associative and sensory. It's almost like cortex kind of violates everything, but probably has its own organizational principles. Okay, I'm sorry to take us off course, but I think for all neuroscientists, this is really important to understand, because obviously the nervous system does a lot of things, but a given neuron is either motor, sensory, modulatory, or some combination of those. So we've got these nuclei, we don't understand what they do, but we're thinking they probably modulate things. So what did we do? We looked at the structures that project to them. And it turns out visual nuclei, and I call them arousal nuclei, which, you know, should make all the neuroanatomists cringe. We're really talking about areas of the brain like the locus ceruleus, which control noradrenaline release, et cetera. They all funnel into the so-called ventromedial thalamus, VMT. So we did a simple experiment, which was to say, what is the role of the VMT in threat responses? And to address that, Lindsay put a virus expressing either HM4D or HM3D into the thalamus. So this allows her to either lower or increase the amount of neuronal activity there in response to a ligand, CNO, which is clozapine N oxide, which alone we believe does nothing. We did those controls. I'll show you those controls. But when this receptor is there, it controls it's now a ligand receptor interaction between CNO and HM4D, which leads to about a halving of the neural activity in those neurons that harbor that HM4D, or an HM3D variety that binds CNO as well, but leads to about a doubling in activity output. So let's see what the result is of lowering VMT. So decreasing VMT activity eliminates tail rattling. You abolish tail rattling. And if we think of tail rattling as a confrontational response, and we want to really anthropomorphize, we'd say the VMT is somehow involved in generating aggression or willingness to stand in threat, and you eliminate tail rattling. And it is significant statistically, but here's the problem, and this should become obvious. There ain't that much tail rattling to begin with. It's not the kind of result that gets you too excited, because you weren't really starting from a place of a really robust result, right? A little bit of tail rattling shown in these little squiggles, it goes away. It's consistent, but it's a pretty minor effect because you're close to the floor when you begin. Now we ask, what is the effect of increasing VMT activity on visually induced threat behavior? And for that, we saw something far more interesting. So for instance, the animal here is just exploring its chamber. The VMT has increased activity. So what you're noticing is that the animal doesn't change its behavior until there's a looming stimulus present. And when that happens, the animal starts doing its tail rattling, and now it's doing it all outside the shelter hide. In fact, it's almost as if the animal's waiting for the threat. Again, we don't know what it's thinking, so we don't know that it perceives it as a threat. And then is tail rattling. Very bizarre response, but suggests that the animal's desire or reflex to freeze or escape is gone, and its reflex to tail rattle, this aggression behavior, and to do so out of the shelter hide has now been enhanced. So almost as if the animal is like beating its chest saying, come on, let's go, let's fight, or at least signaling its presence. Very non-adaptive behavior, if you really think about it. Okay, so now we can look at the actograms here, and what you can see is the amount of tail rattling goes way, way up when you increase activity of the entire VMT. Okay. Increasing VMT activity also promotes these confrontational responses out in the open part of the arena. If you look at, for instance, the amount of tail rattling, that goes way up. And if you look at the amount of running, they're moving around a lot more. Now, they used to run around, the controls rather, run around a lot as well, but they only run toward the shelter hide normally. So if you look at the amount of ambulation that in a control animal, you see that if there's ambulation, it's just to get to the shelter and hide. Whereas in the animals that had the VMT activated, the ambulation is now all over the open part of the arena. So a very non-adaptive response, but I think we can generally conclude that the VMT is doing something at least interesting or intriguing for this visual autonomic interactions. And a key aspect of the experiment that you saw where the animal tail rattled outside the hide is that the moment the visual stimulus was gone, the animal didn't change its behavior. It wasn't just tail rattling like crazy. And that is reminiscent of another set of findings which are worth mentioning, where Dayu Lin, when she was a postdoc in David Anderson's lab, did experiments where she stimulated the ventromedial hypothalamus. That's a nucleus that harbors neurons controlling sexual behavior, mounting and intromission in males, and the lordosis response in females. And it also controls aggression. And she was able to beautifully separate out the activity of those neurons. What she found was that if you stimulate the neurons involved in aggression, when a mouse was in a cage with another mouse, it would try and kill the other mouse. And then if you put the mouse alone in with a glove just filled with water or with air, and you stimulate the ventromedial hypothalamus, the mouse would try and kill the glove. But if you put the mouse in a cage alone with no object or other mouse, and you stimulate, not much changed, at least not in terms of overt behavior. So there needs to be some sort of either visual or other kind of, maybe olfactory driven stimulus present in order to evoke these confrontational responses, which argues that sensory input of some form, I doubt it's just vision for a mouse, but sensory input is absolutely critical. Okay, so you could imagine two opposite strategies for evading threats. One would be to avoid threats. So you could confront. It's one way to avoid threat is to eliminate the threat, right? To fight the threat, maybe to kill it or make it run away. The other is to avoid it. And, we see a range of these responses in nature. I love camouflage. I had cuttlefish in my lab for a long while. We still haven't published the data. The data are very much related to ocular dominance plasticity. And unfortunately, the postdoc left. But I'll someday publish those results. But some animals will hide. Other animals will hide by cloaking themselves with plants, just as military would do. They were stalking. And then, of course, these incredible beasts, the cephalopods, will camouflage in a very dynamic way. That's one way to do it. The other way to do it, of course, is to confront. And there are confrontational responses. Cats have a fear-based response with their ears back. They also have an ears forward confrontational response of making themselves large. You've got the pufferfish. You have animals like this that make it clear to birds, which have color vision, like, you don't want to eat me, you'll die. You see people like this in cities too, that behave kind of outrageously, or sometimes I see, yesterday, when I was at the airport, I saw a person who was dressed like a video game character. But what was interesting was maybe it was Halloween related, but they had so much stuff on them. It was like spikes, like, don't get near me, was at least the signal as I interpreted it, right? It was a very don't come near me kind of signal, at least again, as I interpret it. And then of course we have charging type responses and on and on. So you have these two opposing strategies. What do we see? Well, when you look at control animals, you're mostly seeing avoidance. So that would be freezing or running away, small amount of tail rattling. The HM4D inactivation, as I mentioned, that eliminates the tail rattling. This confrontational response is actually movement outside the shelter hide. Oops, excuse me. And then you see more confrontation relative to avoidance type behaviors when you stimulate the VMT. So the VMT is changing something, but let's not lose ourselves in the anthropomorphizing of this behavior, and ask what is changing. Like, what's changing? We don't know what confrontation is. This is a mouse. We're neurobiologists. I don't know what avoidance is. I just know behavior and autonomic arousal. So that answer came to us by asking this question. I'm an anatomist at heart, and I'm neither a lumper nor a splitter, as they say, but the VMT clearly has multiple nuclei. So what Lindsay did really, was to start putting anterograde tracers like adenoviruses with GFP, and look at the outputs of these different structures and saw, for instance, that the nucleus reunions projected very strongly to the medial prefrontal cortex of the mouse and the intercingulate cortex. We can discuss function in a few minutes. The basolateral amygdala also received and provided inputs to this structure. And then she started to parse which structures were involved in which types of behaviors, because remember, up until now, we're just stimulating or silencing the VMT on the whole. So what she was able to show was that the nucleus reunion's main output is to the medial prefrontal cortex. She did this by anterograde and retrograde tracing. This is retrograde tracing shown here. Whereas the xiphoid nucleus main output is to the basolateral amygdala, a structure that is often associated with fear, but it does other things too. So this kind of anatomical segregation is the kind of thing that makes my heart sing, because I've spent my career basically labeling things in different colors and operating from the belief that nearby structures or layers or modules that take on some sort of segregated arrangement are controlling different types of behaviors. It's not an unreasonable thing to use as a wedge into parsing the nervous system. So next, what Lindsay did was to selectively manipulate them. She used this is a certain kind of canine virus that retrogradely brings Cre, and which of course the Cre recombinase is an enzyme that renders neurons vulnerable to Cre-dependent manipulations. So what she can do is put Cre into the xiphoid by retrogradely infecting from the basolateral amygdala. The nice thing about an experiment like that is that we assume that the xiphoid nucleus has other outputs as well. But in this case, any virus she now introduces to the xiphoid that is a Cre-dependent virus will only infect the neurons that at least project to the basolateral amygdala, although they could very well have projections elsewhere too. What she found was not exciting. The number of running events was a little bit reduced, but it was not significant. So she didn't really see much in terms of an effect of silencing this pathway. However, when she, activating with HM3D, when she activated the nucleus reunions to medial prefrontal cortical pathway, however, what she saw is there was a lot more tail rattling. So it seems like the reunions to medial prefrontal cortex projection is playing a more dominant role. It wasn't conclusive, but a more dominant role. So now we're starting to parse this structure. And then this is important vis-a-vis tools and controls, but we've been using chemicogenetics, but she was able to selectively stimulate just the axons of nucleus reunion cells that arrive in medial pre-FC by putting a blue light over this region, over the medial prefrontal cortex itself, and then rendering a certain number of axons that project to medial prefrontal cortex vulnerable to that blue light, making them more active because they express channel rhodopsin. And in that case, she saw more tail rattling and increased running outside the shelter hide. So it really does seem like it's the axons arriving in medial prefrontal cortex from nucleus reunions that are driving this, what I'm calling confrontational response. So a key question when you do this kind of experiment is, you could imagine that the internal state of the animal is changing when this reunions to prefrontal cortex is in a pattern is an increase or activity is increased. You could also imagine that it completely changes the way that the world seems or appears to the mouse. Maybe that looming stimulus no longer looks like a looming stimulus. That could explain it. So to address that, she did an experiment where she put channel rhodopsin into the VMT. She also did this specifically for the reunions output, and then was able to stimulate the activity of these cells with or without a looming stimulus present. And she could do stimulation before looming, during looming, after looming, and so on and so forth. And I'll summarize what she found here. So keep an eye out over in this panel for when the looming stimulus is on, and then we can relate that to the behavior. So the animal now has a tethered for delivering the light, but the laser goes on, and when that happens, the animal stays out of the arena and shakes its tail. Now, keep in mind, the laser is still on, but the animal no longer shakes its tail unless the looming stimulus is present. This is happening quickly. So laser on, it's not doing anything differently. Then, oops, let's just go one more time. Laser goes on, stimulus appears, the animal starts tail rattling. In fact, it runs underneath it and tail rattles. Laser stays on, but stimulus is gone. It sits there pretty passively. Okay. So again, this tells us that the activation of reunions to medial prefrontal cortex, or the VMT on the whole, doesn't change the behavior of the animal. But if a stimulus is present, if a threat stimulus is present, the animal will change its behavioral output and become more confrontational. This is quantified here. The number of tail rattling events and running events is again statistically increased. So that doesn't rule out the possibility that there's a change in internal state. And in fact, there is. So if an animal is just sitting in its cage, you're looking at pupil size. Again, increases in pupil size are a reflection of increase in sympathetic tone. Activating VMT or reunions alone increases pupil size, also increases heart rate. It also increases breathing rate. This is just an apparatus that allows us to measure these. Hopefully these will become less invasive and odd looking in the time goes on. This was a fun experiment that I love because I love it when the human data and animal data converge. So the question was, is the activation of VMT a positive, negative, or neutral experience? And we did a simple place preference test where, again, we, meaning Lindsay, activate the VMT anytime the animal crosses to this side of an arena, not when it crosses to this side of the arena. So it's a real-time place preference test. And what we found was that animals love having their VMT stimulated. In fact, later we showed they will work to have their VMT stimulated, which is wild because this is a confrontation response. Or maybe not so wild. You know, we just spent 2 years with people, seem like there's a lot of confrontation, a lot of fear too, but a lot of confrontation. And there's a result that I'll share with you, and you can look up the paper if you like. It's a paper published in 1963, 1965, and again, a few years later, basically the same data, published 3 times, once in Science, once in PNAS, as one used to be able to. But the result is really interesting. It's an experiment from a guy named Robert Heath, who is not a character that I would want to meet, but he was a neurosurgeon, neurophysiologist who put stimulating electrodes in the brains of human beings at a time when you could do this, and then asked people to stimulate different areas of the brain, ask them subjectively what they experienced, and ask them which areas of the brain did they like to stimulate. And he put electrodes kind of all over the place, but he put some in the midline thalamus, central median nucleus in particular. And people would stimulate certain brain areas and say that made them feel drunk. Other brain areas, and it would make them feel sexually aroused. Other brain areas, it make them feel giddy. Other brain areas, it make them feel sad. And certain brain areas would make them feel frustrated and angry. And that was the central median nucleus. When you stimulate the central median nucleus in humans, and this has now been verified with modern tools, humans get frustrated and angry. They don't go into a rage, but they get angry. And if you ask people, at least the subjects, what is your preferred stimulation site? They all, meaning all 3 of them, it's not very many subjects, say the central median nucleus, it feels kind of good to be angry, which is surprising to me, but was prompted Lindsay to look at the outputs of these structures. And this is including the supplemental data of the paper, but I'll just give you the takeaway, which was that there's a collateral to the ventral tegmental reward pathways for these confrontational responses. Confrontational responses are rewarded neurochemically. And, you know, I think about human behavior very differently now. Again, I'm gleaning from one study on 3 patients and a bunch of mice, but, you know, I see people and I know people that seem to like to argue. There's this kind of like this pleasure in arguing. I think there's actually is it a play or a skit about this where you could pay someone to argue? I don't know. Is it right? So, you know, there's, you know, and then there's Twitter, right? Where now it looks like there's going to be an $8 a month fee to argue. You know, it's not totally surprising once you understand the context of some of this stuff. And I think there are some rewarding circuitries in there that underlie this. In any sense, mice do this. So I love this image. So I wish I had shot these, but I didn't. So let's go back to our question of visual autonomic interaction. How precise is this, right? I mean, the circadian thing is like if you see light in the middle of your circadian night, so for a mouse that's in the day and for a human that's in the middle of the night, it tends to increase your activity, reduce melatonin, and shift your circadian clock. That's arousal. That's the sleep-wake thing. But what about something more sophisticated? That's sort of like a yes-no kind of binary system. Light equals arousal, lack of light for a diurnal species. Light equals arousal, lack of light equals lack of arousal. But for animals in the wild, there's more nuance than just incoming birds. And in fact, sometimes a bird will do this. It'll just fly over. And if you're the potential prey of that bird, movement actually becomes non-adaptive. This goes back to our lion example at the beginning. So this is an experiment that Lindsay did where she puts a mouse in a chamber and then she provides a visual sweep as opposed to a looming stimulus. And every time the animal just freezes. Now, we've also taken away the shelter hide. That's important. If you keep the shelter there, they will actually use it every once in a while if they are close to it. But watch how early this mouse freezes. Watch for the appearance of the stimulus. It's almost like the mouse knows it's coming. But if you look, it's about 100 milliseconds after the appearance of that little dot in the corner is incredibly fast. Remember, there's a colliculus mediated behavior. Wow. I don't know. Every time I see that, how can it do 100 milliseconds? Amazing. Okay. So the VMT activation promotes confrontational behaviors if it's a sweep or if it's a loom. Now, this tells us further that, yes, there are changes in the animal state, but that it still can detect the difference between looms and sweeps. It's not changing vision in some fundamental way. And this is important because if you recall, when you stimulate the autonomic system any which way, caffeine or fear, the pupils get bigger. And when pupils get bigger, optics change. And when optics change, vision changes. So this is important in terms of a control. We also saw this with olfactory odors. We do not think that the ventromedial thalamus is uniquely controlling autonomic visual interactions. You put a mouse in a chamber, divide it in two, you put fox urine or some sort of predatory urine on the other side and they avoid it. But if you activate the VMT, they will approach it far more. They'll confront it far more. Typically, they would go over, sniff it, and then back up, and then not go back over there. They might explore it a little bit. But if you activate the VMT, the animals will confront the odor of a predator. And likewise with visual loom. So in every case, activation of the VMT and reunions in particular, we find, is making animals more confrontational. Okay, so I want to just let that sink in. And I know we have we are going to get to some questions in a moment, but I want to just share with you one more result and I'll go through it a little bit quickly, but that tells us where all this is converging and how this relates to vision. Because unfortunately, or fortunately, depending on how you look at it, I can't drop my vision hat. A neural circuit that regulates internal state should promote flexible behaviors. We're not talking about reflexes, right? If I step on a tack, I retract my foot. This is classic, monosynaptic reflex stuff that you learn in Neuroscience 101. That's a reflex. Of course, it hurts. It's going to change my internal state, but internal state isn't driving the reflex. The reflex is more or less happening in parallel with or governing internal state. Induces changes in autonomic arousal. We know that the overall milieu of the animal is different. It's more alert. It's less likely to take a freezing response or an escape response. Again, freezing or pausing is the lowest autonomic response. Escape is the next one. And then it turns out that confrontation is the highest level of autonomic response, which kind of makes sense if you've ever gone into a conversation and you're not Ed Ruthazer, so you're not unbelievably calm like me, and you tell yourself, I'm going to be really calm no matter what they say. I'm not going to argue. I'm just going to listen. I'm just going to pause and I'm going to listen. Or if I don't like what they say, I'm just going to walk away. And then 20 seconds later, you're yelling back, right? It's the highest level. Confrontation is the highest level of autonomic arousal. And we forget this because we think of freezing as paralyzed in fear. It persists beyond the triggering stimulus. Now, gets to a bigger discussion about states versus emotions. David Anderson has talked a lot about this, and Ralph Adolphs as well. States have intensity and persistence that relates to autonomic arousal. Emotions are a subcategory of states, not the other way around. And David's really the expert on this, but I find that to be a very useful framework rather than think about states as, emotions driving states. It's the other way around if you really think about it. And then these neural circuits governing state, they have a predictive power. They can really tell you something about what you might expect to a given stimulus, right? If you have low levels of autonomic arousal and you have a certain stimulus, you predict pausing. If like the sweep, if that's adaptive. If you have higher levels of autonomic arousal, the same stimulus will evoke more movement. And then, of course, if you have highest levels of autonomic arousal, you get confrontation. So this is the way that we think about it. Okay, now we did some experiments to show that indeed the VMT, has elevated levels of firing during threat behavior. But I should mention, if you keep giving a stimulus, this looming stimulus, the animals attenuate and they started ignoring it. They learn that you're crying wolf. They're not going to get eaten. But what's interesting is the VMT actually starts to reduce its activity from day 1 to day 3. Day 1, they're freezing a lot. Day 2, not at all. And day 3, they're not freezing at all. And the activity in the VMT falls accordingly. So the VMTs does really seem to be driving something important there. So it tracks to internal state, not to looming. Okay, so this says part 2, but I'm going to be very brief about this. I couldn't let Lindsay leave the lab until I understood how the visual system and this set of midline nuclei really link up. Because right now we're playing with midline nuclei that talk to prefrontal cortex and basolateral amygdala, and we're doing visual stimulation, but the olfactory stuff does it too. And, I was starting to worry about myself because there wasn't ganglion cells involved. So what Lindsay did, and she published this work last year, was before she left, she started tracing out of the VMT to these visual areas. And one of the more interesting areas with this thing called the ventral lateral geniculate nucleus, which in primates is sometimes called the pregeniculate nucleus and is sometimes also called the perigeniculate nucleus. And in cats is sometimes called the geniculate wing. And no one can agree. And if you talk to Murray Sherman or the late Ted Jones, who was a friend of mine, a gruff guy, but a very knowledgeable guy nonetheless. I'm sure Bernice, if you probably interacted with him at some point, well, that's true. So what you'll find is that everyone argues about these things, but the ventral geniculate sends direct projections to the VMT. And that's interesting because the VLGN is not very well understood in terms of its function. We know it receives input from melanopsin ganglion cells and has circadian functions, but there's a lot of other ideas about what it might do based on anatomy. It's got a lot of subdivisions, et cetera. So what Lindsay did and essentially showed, and here I'll speed up, is that retina projects to DLGN, to visual cortex. Of course, this was known. Melanopsin ganglion cells project to the VLGN, and then the VLGN basically talks to everything. It's not just a structure involved in circadian timekeeping. So she did a bunch of experiments retrogradely labeling from the VMT to the VLGN to put Cre in those cells using viruses, so using a canine virus, then put a Cre-dependent virus into the VLGN, and then a helper virus, and then identified the ganglion cells that project uniquely into this pathway. So now in red are the cells that project to the VLGN, only to the cells that connect to the VMT. That's the way these tracing experiments go. And then this is really what matters, which is that most of the cells are non-melanopsin cell types. So here we have a pathway from retina to ventral geniculate to VMT that is unique from the circadian timing system. She then did the same sorts of experiments you heard about before, manipulating only those cells in the VLGN that project to the midline thalamus. And it turns out these are of the GAD expressing or GABAergic variety. Did this a number of different ways. And I'll just show you the behavior here. It's a little bit dark on purpose because that's how the mice need it in order to get good behavior out of them. But basically, I'll let this play for a bit. This is sped up. If you inactivate the VLGN projecting to the midline thalamus, the animals, I'll do that one more time. The animals freeze the entire time for minutes. Whereas if you activate them, they run around a lot. This is a wild result if you really think about it. The animals are freezing from the first loom until almost a minute and 30 seconds. Let me just show you the actograms, and it's just apparent, just the pink is freezing. So you activate these GABAergic cells in the VLGN, you turn the looming stimulus on and they just freeze, but they're not somehow impaired as an animal, because when you turn it off, except for this animal, who was just petrified apparently, or just liked it, just kept going. Whereas if you activate, you see something a lot like VMT activation, where you're getting tons of tail rattling and at least far less freezing. Okay, so that was fun for us because it identified a new role for the VLGN. I'm sure it has many roles. And then she really started to go to town on all the tools. And Lindsay was they don't respond to looming. These cells in the VLGN don't care about looming. They're not looming detectors. They are luminance increment detectors. They really care about increases in luminance. And you can see this by, stepping a stimulus brighter. This is done by imaging, by calcium imaging cells within the VLGN in vivo. You can see they increase their output, and so on and so forth. And so she then went straight to a behavioral experiment to mimic this kind of thing, right? Animals have to make good decisions about whether or not to leave holes or stay in holes in order to forage. And she developed what she calls the whack-a-mole assay for light avoidance. And I'll just quickly show you this, which is that typically animals if there's two chambers above and below, if it's dark on top, they'll leave and move around a lot. If it's light on top, they leave far less. They're smart. It's quantification. When it's light in the top chamber, they stay out of the top chamber. VLGN neurons control this. So if you inactivate VLGN neurons, the latency to enter the top goes up. If you look at how much time they spend in the light and you inactivate, they spend a lot less time there. If you activate, they spend more time up there. So it's a switch, but it's a switch that's communicating with the midline thalamus, and it's a switch that's receiving input from the retina, and we know it's retinally driven. So it encodes something related to light intensity or relative light intensity. And again, fear is in quotes because we don't know what the animal feels. So to summarize, this is the set of neural circuits that Lindsay identified. The xiphoid nucleus has outputs to the basolateral amygdala, seems to be involved in tail rattling responses in some way, but not in a major way. It's more involved in freezing and avoidance. The reunion projections of prefrontal cortex are modulating the decision or the state that allows for confrontational outputs. The retina is projecting the VLGN, which is then feeding these structures, in particular reunions. Don't have too much time to go into that today. And as a consequence, environmental light can serve as one more stimulus by which to adjust the autonomic arousal of these animals. So overall luminance in the case of the VLGN, the looming stimulus specifically, and how the looming stimulus information gets access to the midline thalamus, we still don't know. Presumably it's from the colliculus, but we don't know. And that's an interesting idea. But I think what Lindsay did, at least partially, was, at the beginning of all this, we knew there was a looming stimulus. We knew that animals could adjust arousal based on circadian time scales. But her goal was really to link vision and internal state on these faster time scales. And she started to parse these thalamic circuits. And she identified this retinothalamic projection for biasing behavioral decisions to luminous variant environments. This brings us all back to some high-level questions that we can just ponder on our own or during the Q&A. Just, what are the neural circuits that integrate vision? We've identified at least some of those, but we don't really know. And so let's kind of leave this open for discussion. There's a lot there to parse in terms of reactions to faces and symbols. And, for instance, certain symbols of certain groups evoke a very strong response. Flags, et cetera. Faces of politicians and things, especially nowadays, really evoke a lot, right? And you think about this in terms of avatars, and you think about it in terms of online interactions. And, sure, in part, it's based on what someone said or did. In part, it's also based on just their face can evoke all sorts of categories of responses. And once your internal state is shifted, if nothing else, the data that Lindsay showed indicate that shifts in internal state dramatically alter our likely both behavioral outcomes, but presumably psychological outcomes too, and do all that without actually altering our perception of the stimulus, the visual perception itself. So I just want to thank people. I tried to mention her name as often as possible because she was the one that did all this work. She got some assistance from these other two folks who are terrific. Melise did the human work. We can talk about the human work some other time, perhaps. That's an active area in my lab. More than half of my lab is working on humans. Hee-Kyung does observational fear in the visual system. Suraj is the one working on regeneration, and Gary is our lab operations manager. And I just want to thank you all for your time and attention. And I think I'm at least not over the end of the hour, and I'd be happy to take questions. Thank you.
Yeah, thank you so much, Andrew. That was great. So it looks like there's already a question from the audience from Gina Kemp. Gina, do you want to just unmute yourself and ask?
Yeah. Thank you so much for the talk, and thank you so much for organizing it. I do have two questions. One is big picture, and I think it's the most interesting one. Just in the interest of time, I'll just stick to it for now. You've mentioned that you work on humans, and that makes me extrapolate that you are interested in the translational aspect. And I do appreciate quite a lot, as somebody who's very, very much interested in this field, that you don't go into this classification of behaviors and the muddy water of saying which is what based on physiological, because it's really like a circular argument, right? You say this is fear or anxiety or aggression because this is the behavior, and this behavior because it's fear, anxiety, aggression. So I'm very appreciative, and I really enjoyed that preface and this theme throughout is to stick to arousal. But I do want to pose a question that I think about quite a lot, even though I do agree with this idea that we should stick to the physiological output as a way to objectively operationalize psychological constructs instead of going into, let's say, the linguistic description of it, and that there is this dissociation between language and the physiology
Gina,
part. However, the question is how would you approach then in terms of translation that humans go to therapists or psychologists or psychiatrists because of something they can express consciously with language? People will not go to therapists because, let's say, they have arousal in the ventromedial, in the basal lateral amygdala or other parts of the brain. So that's really the question. Thank you so much.
Yeah, sure. No, it's an important question. So there are a bunch of directions that we're taking this in humans, and forgive me for not getting to that, but it would have put everyone to sleep. I was joking. My podcast accomplishes nothing else. It might cure insomnia after all, because the episodes are very long. So here's the deal. So in humans, we have a VR setup that's very much like the mouse setup, but we can expose humans to specific things that they don't like. Looming great white sharks, height stimuli if they're afraid of heights, dogs if they're afraid of dogs. These stimuli for most people are not a problem, but if you have an active fear of something, they're really a problem. And so we can reliably evoke autonomic arousal with negative valence. So that's good. And in fact, Melise Yilmaz really deserves credit for basically building that arm of my lab at a time when VR was very nascent. She figured out all of that. She worked with dog trainers to have dogs attack people and film in VR with 360 cams. It's a pretty cool setup that is safe for people, right? It's not natural in the sense it's not real world, but then again, we can only do so much before people start getting injured, right? So, and we've had only one sort of accident in the lab of somebody really trying to flee inside of the VR. So VR can, about 90% of people can get what's called presence inside the VR if you get it, if you build the stimulus right. What have we learned? Well, we've learned that indeed humans show the lowest levels of autonomic arousal to the freeze is associated with the freezing response. The escape responses would be a slightly higher levels of arousal, and that confrontation is something that can be trained. In some ways, this has already been done through cognitive behavioral therapy, right? But in cognitive behavioral therapy, like, let's say I'm somebody who's afraid of pens, for lack of a better, but so like, I happen to love these V5s, these Pilot V5s. I adore them because they write well and they're inexpensive, and I lose things all the time, so it's perfect. But let's say we're afraid of it. Cognitive behavioral therapy would have me think about it, then draw it, then not with a V5, then look at it from across the room, then approach it, then pick it up. And the goal would be to bring my levels of autonomic arousal down, right? The approach that our data suggests is that there are instances in which getting people into higher levels of autonomic arousal might allow them to actually confront the fear more readily. And we're talking to psychiatrists about how you might incorporate that. This would be sort of shotgun exposure therapy, not to actual shotguns, but throwing people in the deep end of the water to teach them how to swim, kind of thing. There are ethical issues, of course, around that. We have to be careful in what we extrapolate to. The other thing that I think is perhaps equally or more interesting is, there's a growing interest in creating sensors for autonomic arousal. And some of these sensors are very small. So there's a group at Illinois, Urbana-Champaign, that has engineered a little Band-Aid that's 12-hour Bluetooth. It goes on your clavicle, and it can measure breathing, heart rate, and cavitation. But it can also measure, this is amazing, it can also measure changes in vocal tone across the circadian schedule, how many arguments you're having, how much literally negative language, how much your internal arousal state is based on what you're saying. And it's actually being used by a group up at University of Washington to try and predict suicide tendencies for people that themselves are not aware that they're veering towards a suicidal depression again. So these are individuals that are prone to suicidal depression. It's designed to catch people's signature of autonomic arousal that anticipates a suicidal episode before they even realize they're becoming suicidal. In other words, there is the possibility that AI of ourselves can teach us more about how we are going to feel in terms of general states than we can address subjectively. I find that very exciting. It's going to require a lot of people far smarter than me and far better at engineering than me, which isn't hard to do, but there's some great engineers out there that are figuring this out. And you kind of think, oh, wow, AI, they're really going to be peering into our system. The insurance companies are going to want, there's all this paranoia. But I love the idea of a sensor that says, Andrew, you've been really pushing pretty hard for a day or so. You might feel great, but your autonomic system is kind of headed toward, your sleep is a little more fractured, or your autonomic system is heading for you're going to be prone to illness. Now, we do a little bit of work on sleep in humans. We have people wearing autonomic sensors, and we're measuring sleep. We're also paying attention mainly in those studies to their patterns of breathing. Are they predominantly nasal breathing or mouth breathing throughout the day? Let's just face it, there are great data from colleagues of mine at Stanford showing that if you're not talking or eating, being a nasal breather is best for your nervous system, your immune system, your brain. There's a beautiful data that even on inhales, your sensory detection is far better of visual stimuli. These are published in great journals, by the way. Noam Sobel and others are doing this. Like, there's something, it makes sense ethologically when you start thinking about inhales versus exhales, but are you nasal breathing? Are you regulating your internal state, or are you veering towards kind of burnout? And I think I'm not a fan of discarding a subjective data. I think people describing how they feel with language is very important. But I also think that, especially in these extreme conditions of suicidal depression, or where people are veering towards insomnia, or people who are recovering from trauma need additional rest for invoking neuroplasticity and things of that sort, I think it's going to be a really powerful tool. And so autonomic sensing and coupling that with what's going on in the sensory environment, I think is going to be an interesting area. And a lot of these non-invasive tools, I think, are going to give us a lot of information about how to change our behavior. For instance, I'll just give you one simple one, and I hope someone will engineer this. I would love a pair of glasses. I actually wear readers now. I'm just not wearing them. I would love a pair of glasses that in the morning would change, the frames would change color if I got enough photons. And then in the evening would change color if I was getting too many photons from screens, right? So I could have just kind of a readout of, am I getting enough daylight or light, or am I not getting enough? As opposed to me having to walk around with my phone and a light meter, or listening to me blab on my podcast about how much time you need on a cloudy day. I mean, how cool would that be? You're like, oh, it's sort of like measuring your steps. People are measuring, did I get 10,000 steps today? Why not did I get 1 million photons before noon? I think that stuff is so easy to do and is transformative for well-being, but transformative because it puts our autonomic nervous system into the correct state for the general circumstances we're in. None of this stuff is that complicated to engineer. And rather than people walking around, no disrespect to blue blockers, and the blue blockinistas are going to come after me, but like wearing glasses to try and suppress the light in your environment might make sense in a certain context. But in general, it's pretty simple. Get a lot of light during the day and limit the amount of light at night. But how do you know what enough is? And I think sensors like this, or sensors that are measuring your overall, how many episodes of real stress did you have today? I mean, think about the implications for the cardiovascular health arena. This is huge. And of course, you can have heart rate increases because you're happy. And we know that that helps your nervous system and your cardiovascular system. If you have heart rate increases because you're unhappy, it's the opposite, but that's pretty easy to parse. So I'm very excited about autonomics, genomics, autonomics. I don't know.
That's
I'm not going
to. Yeah. Well, one of the questions that I just saw pop up is something I wanted to ask too. From Alexa Brown, she asked, are there any sex differences in the expression of confrontation versus avoidance?
Not that we could observe, nor did we see alterations along the changes along the estrous cycle for these mice. There are interesting sex differences in Hee-Kyung Jung's experiment. So Hee-Kyung came to my lab from the world of really cool experiment. She was in Jeremy Dason's lab, and she studied the motor neurons that control, these animals that like stingrays that flap along the ocean floor, but then they actually can pedal and kind of walk. They can do a kind of, and so these motor neurons, they can switch. Anyway, beautiful work. When she came to my lab, she studied much in the same way that Jeffrey Mogel in your arena up there says this for pain, observational threat. So she exposed animals, saw a loom and froze, and then animals were watching the other animal, and they would undergo some observational fear response. So this is kind of social contagion or emotional or state contagion. And there you see interesting sex differences where same sex seem to modulate their behavior according to same sex more than opposite sex. You have to do a little bit of teasing out of these data. It's not super obvious. They don't fall out into strict bins, but we do see sex differences there. And yeah, unfortunately, those data aren't published yet, but I'm not hiding anything. It's just that's the bulk of what we discovered, and it involves an interesting structure I've always been fascinated by. There's a subdivision of the claustrum that's involved. And I have to say, and maybe Ed, you'll chuckle too, and I don't know, Bernice, what your thoughts are, but for years I was told that the claustrum, the pulvinar, and the midline thalamus were sort of the graveyards. I think Mort Mishkin told me that the pulvinar was the graveyard of neuroscience. Nowadays, everyone's excited about pulvinar, claustrum, all these structures again. But it's amusing to see how the times have changed. I think it's actually gotten better. I think now, every area of the nervous system and body is fair game for study as long as you're doing good experiments. So that's exciting. When I was growing up in science, it was a little bit more like a John Hughes film. You had to be in one camp. Now it's a little bit more all-inclusive.
Yeah, Helen Shirk was a kind of visionary, I think, in many ways. It looks like there's a question from Noor Shaheen. Noor, did you want to ask your question?
Yes, I sent it on the chat but I'd rather ask it in person just to clarify myself because it might be a bit too wordy. So I know that from what we have seen, when the VMT is activated, there is an induced, like a higher proportion of confrontational behavior. I was wondering how lasting this effect is. So you mentioned that it is lasting even after the VMT is activated, but I was wondering if this can be transformed into some kind of learning protocol where there is some kind of training with VMT activation for a certain period of time. And then after this period of time, the mouse would exhibit a learned confrontational behavior towards looming stimuli or such things. And if so, if this is possible, I was wondering how applicable this could be in terms of treatments of phobias or disorders that involve people learning to be more confrontational rather than avoidant. I hope my question was clear. I don't want to like overcrowd or anything.
No, your question was exceptionally clear, and it's an important one. So the short answer is yes. There are ways in which you can evoke increases in autonomic arousal. This gets a little tricky in humans when you have someone who has an anxiety or a panic attack disorder, because obviously they don't want to feel that. But in terms of phobias and treatment of generalized anxiety, you know, we hear generalized anxiety as a disorder, but there's also generalized confrontation or approach behaviors. And the way that this is accomplished is actually to pick a stimulus that someone can tolerate. They tell you they can tolerate it, that increases levels of autonomic arousal, and then transplant them into a situation where they are, you know, forced to deal with the thing that normally would be too much for them, right? It would be overwhelming for them. Evoke freezing or escape or just a kind of a collapse of mental functioning. And I want to be very clear before I continue that when I say confrontation, I mean adaptive confrontation. I mean, there is something really pathologic about a mouse that's being loomed on by a predator that is there slapping its tail saying, come and get me. This is akin to the these horrible videos on YouTube of these kids, you know, doing parkour in cities, you know, jumping between buildings and doing pull-ups on the sides of buildings. You just go, oh my gosh, I can't believe, like, of all the material I think should be taken down off the internet. And I'm not big on, you know, I'm more or less I am for free speech. I'll just say, like, I believe in free speech, right? Which is not to say you can say anything. It's just saying that for nonviolent, you know, generally expression, great. But, you know, there's so many things on the internet that are showing people doing absolutely non-adaptive behaviors that people see and then they go try and it doesn't work out well. So I think that when we say confrontation, we mean adaptive confrontation. And I think that, you know, there's not to draw everything back to the podcast, but there's sort of a growing trend now towards exploring autonomic arousal as a continuum and as a tool. And so I'll give you an experiment. So in our laboratory and in a collaboration with a guy named David Spiegel, who's our associate chair of psychiatry, David's lab studies medical applications of hypnosis. So this is, you know, everyone's familiar with meditation. It's a self-directed perceptual shift towards either thinking about your thoughts or thinking about your breathing or focusing. Okay, we all know what that is. Good data support that it can be useful. Although I should mention, people who tend to be kind of in their head, quote unquote, don't do well with eyes closed meditation. It tends to make them more in their head, whereas they would do better with an eyes open, external focused meditations. There are good data on this now. People who are very out of touch with their physiology, the sort of type A personality that the cardiovascular community defined as people that are like, go, go, go. They don't even realize that their blood pressure is high. They would do better with some more interoceptive type behaviors. Okay, why am I telling you all this? Well, it turns out that if you take people, and we've done this, and you give them a way to self-stimulate their own adrenaline, and we do this by encouraging them to, we don't force them, but to doing cyclic hyperventilation. So they essentially hyperventilate, which dramatically increases adrenaline. If you hyperventilate for 15 breaths, you will feel warm. That's adrenaline. That's essentially what it is. Then you put them into a confrontational state, you put them into a state in which confrontation is an option. They actually seem to manage better in terms of adaptive decision-making. And we do this by giving them it's not a tough game, but it's a cognitive game that they have to do while paying attention to some visual flankers, et cetera. It's in VR, so the contingencies are meaningful, and they get rewarded and so on. And what you find is that some people are having high anxiety responses under normal conditions because their autonomic arousal is a little low, they're mellow. And then you put them into a state where they shift and it feels like a lot. So what we're doing here is we're using a self-directed cyclic hyperventilation, putting them into a state in which autonomic arousal, the baseline is a little bit higher. And then for whatever reason, the delta, even if it's the same to a higher levels of autonomic arousal, doesn't feel quite as jarring and they're able to make better decisions. So we're not like putting them into panic intentionally. What we're doing is what we think is happening is the following, is that when we think of autonomic arousal on a continuum of, well, okay, you could be dead, no one wants that. You could be in a coma, no one wants that, but that's very low autonomic arousal. You could be deeply asleep, you could be lightly asleep, right? There's a range in there too. You could be alert but calm, you could be drowsy, alert but calm, you could be panicking, panic disorder, you could be full-blown panic. It's not a linear continuum. At every node, and we don't even know that they're nodes, it could be a continuum, but at certain locations, imagine that continuum is like a pipe, and at certain locations, the pipe is thicker. And at those locations, the behavioral choices are actually greater than it is when you are at a lower state of autonomic arousal, but the pipe, and here I'm talking about the continuum as a pipe, is thinner. And so you can model this as like behavioral access to thoughts about choices and access to the belief that those choices are possible. And why am I, then this gets very psychological, but as Spiegel would say, it's not just about the state that you're in, it's also about whether or not you had anything to do with getting yourself there intentionally. And this is really interesting because if you take people who are just terrified of public speaking, but you bring them into the same level of autonomic arousal that they experience when approaching the podium, and you can do this experiment virtually or in the real world, what you find is that they are comfortable with the state. There's something that doesn't work for them about the state and the context. And so you can teach them how to generalize from one state to multiple contexts. This is so instead of very narrow desensitization, what you're talking about you're talking about broad, sort of I don't know if we call habituation, but you're expanding their the range of experiences under which they can tolerate this higher levels of autonomic arousal. And the other way to put it is that sometimes higher levels of autonomic arousal are better. They're more adaptive. Sometimes they're less adaptive. And so if there's any hope for psychiatry in terms of this stuff and treatments, psychology and treatments, I do think it's going to depend on having a very clear nomenclature so that we're not spending 10 years arguing about the difference between stress and anxiety. What a waste of a bunch of good lives. That we're not arguing about the distinction between adaptive and non-adaptive. I mean, I do think that biologists need to kind of punch their way in a little bit and say, hey, let's just like agree on some terms. And then the psychologists, I think, are great to bring to neuroscience because then the neuroscientists have to deal with the fact that like not everything happens in an experimental context and that thoughts matter. So I think the two really need to marry, the fields need to marry in a way that merges physiology and the subjective stuff. Sorry if that was a little bit of a Baroque answer, but I hopefully got.
It definitely like broadened my perspective on this. So thank you very much. And thank you also for delivering a very clear and accessible topic, like especially for people who are not so familiar with this. It was very clear. It's very much appreciated.
Thanks for your feedback.
Thanks again. So we're now 20 minutes into our fantasy wine and cheese. So
I'm sorry. Okay. And it's later there.
It's great. It was really wonderful. It's so pleasant to listen to you talk. And so I really do hope we'll get you back again before too long. I want to just thank you, and I hope the audience too, to thank you for a great presentation, something that's really changed my way of thinking about midline structures in the brain and in the thalamus in particular. So thanks for that. And yeah, thanks to the audience for coming and asking great questions and for support.
Yeah, thanks so much. And thank you, Bernice, Dr. Grafstein, for all your incredible work. This was a real honor for me. And I am very familiar with your work, and it's I teach it, and it's a front end in all my talks on regeneration. And so thank you. And thank you, Ed, and the organizing committee, the students, and everyone else. And then also thank you, Ed, for not poking fun at me. Ed has a history of he's got great jokes. He's one of the funniest people I know, and I really appreciate that today I wasn't the target of any of those jokes. So thank you, Ed.
I don't know. I think the students won't recognize the person you're describing, but sounds good. All right. So have a good day, and thank you so much again. So everyone, take care.
Great. Be well, everyone.
Thank you so much. Thank you.
Cheers. Take care. Bye for now.